Hello everybody. Dear students, hope you all are fine. Welcome to another lecture of Biochemistry. You are listening to your very favorite Dr. Sandeepan Sengupta, who is an assistant professor in microbiology. In previous classes and lectures, we have been discussing about various fundamental aspects of protein structure. We have gone through different amino acids, the building blocks of proteins, 20 naturally occurring amino acids, the special features exemplified by each of these naturally occurring amino acids, peptide bond and the biochemical features and structural features and geometrical features of peptide bond, about various levels of protein structure, and of course about many more things. So as we have already studied, to recall, you all know that there are 4 different levels of protein structure: primary structure, secondary structure, tertiary structure, and quaternary structure. And what are proteins? It's all known to you that proteins are nothing but the polymers of the monomeric units called amino acids, which are linked together by CONH amide bonds or the peptide bonds. So the primary and the secondary structures of proteins are the 2 basic levels of protein structure, while tertiary and the quaternary structure are the higher orders of protein structure. In our previous classes, we have discussed in very detail about the aspects of primary structure and about different elements or different forms of secondary structure of proteins. So by primary structure, we mean to refer to the linear sequence of amino acids in a protein which are linked together by CONH peptide bonds. By secondary structure, we mean to refer to local conformation or the structure that is adopted by not by entire protein but by a part of the protein or polypeptide. So now today our topic of discussion is supersecondary motifs and structural classification of proteins, and gradually discussing about the supersecondary motifs we'll move on towards the next high level of protein structure, that is tertiary structure of proteins. Although as in our previous lectures we have discussed a lot about few fundamental aspects of tertiary structure, about the classification of proteins, grouping them into 2 classes, fibrous and globular proteins. We have studied the structural aspects of many fibrous proteins and so on. So it's expected that you all are having a primary knowledge about the tertiary structure. Now let us first study the differences between the secondary and the tertiary structure of protein. What do we mean by secondary structure? By secondary structure, we mean to refer to local conformation adopted by a part of the protein or polypeptide. But what is tertiary or still higher structure of protein, or the 3-degree structure of protein? By tertiary structure, we mean to refer to the spatial arrangement or the 3-dimensional arrangement of all atoms present in a polypeptide chain. Hence, tertiary structure is a long range aspect of the amino acid sequence and is a broader aspect of protein structure. So, broadly speaking, while secondary structure considers the relationship or considers the spatial arrangement and the interaction between the amino acid residues which are close together and nearby placed in a polypeptide chain. But in tertiary structure, in what we consider is the long range aspects of the amino acid sequence. So the interactions between the amino acids which are not nearby but are quite distant or distantly placed in the polypeptide chain and are present in different elements of secondary structure or in many different polypeptide chains are considered in what is called the compact or the globular structure of a protein, called its 3-degree structure or its tertiary structure. The location of bends in the tertiary structure of a protein or a polypeptide and the direction and angle of these bends is determined by the number, by calculating with the number and the location of specific bend producing amino acid residues present in a polypeptide, while considering the tertiary structure of the polypeptide, like serine, threonine, glycine, then alanine, proline, etc. Now, in order to have a thorough understanding of the tertiary structure, let us start from supersecondary motifs, and starting from supersecondary motifs, we'll gradually move towards tertiary structure so that it become the concepts or the basic fundamental issues become crystal clear to you. So what are supersecondary motifs? As you all know that there are several different forms of the secondary structure of proteins, like alpha helix, beta pleated sheet, beta turns, loops, omega turns, gamma turns, etc. Now, in supersecondary motifs, these elements of secondary structure are combined together. So the combination of the different elements of secondary structure are first placed into groups or classes called supersecondary motifs. So supersecondary motifs are nothing but the combination of the elements of secondary structure and the adjacent connections between them. They consist of alpha helices combined together or alpha helices combined along with beta pleated sheets and so on. Now next to this, what happens? The thing which happens is that the adjacent elements of secondary structure along with the supersecondary motifs are further compacted and folded to form a globular lobe-like structure called a domain. And what actually is what is a domain, or how many domains are present in a polypeptide? Actually, every peptide chain of a polypeptide is made up of multiple such domains grouped together. So a domain, a domain is formed. What is a domain? Is nothing but is formed when the elements of secondary structure and the adjacent supersecondary motifs are further compacted and grouped and looped to form a globular lobe-like structure. So there are multiple domains present in every peptide chain of a polypeptide, and the spatial relationship between these domains present in a particular peptide chain of a polypeptide adopts a form of structure meant for the entire polypeptide, what is called the tertiary or the 3-degree structure of the polypeptide. Hence, by tertiary structure, we mean to refer to the spatial relationship and the arrangement, the relationship between the domains and the arrangement of domains which are all together presenting a single peptide chain of a polypeptide. So there are different types of proteins containing different peptide chains, like monomeric proteins are made up of a single peptide chain. Multimeric and oligomeric proteins have many different polypeptide chains. For a monomeric protein, which consists of a single peptide chain, the tertiary structure and of the peptide chain, that is the spatial relationship between all its domains in a particular peptide chain, is the tertiary or the 3-degree structure of the entire protein. But for an oligomeric protein or for a multimeric protein, which is made up of several peptide units, what happens? It's quite expected. You all must be knowing. Guess what happens? What the thing which happens is that for an oligomeric or multimeric protein made up of multiple peptide units, every peptide unit will have its own 3-dimensional structure and spatial relationship between its domains present in every peptide chain. Thus, for an oligomeric protein, every peptide chain of a polypeptide will have its own 3-dimensional or tertiary structure. Now, let us come and peep through a bit, peep into the more detail of the structural features of a domain domain present in a polypeptide chain, whereas the domain is actually 2 to 3 nanometer and consists of 100 to 200 amino acid residues, and as a result of further compacting, folding and looping of super secondary motifs and the elements of secondary structure in a domain, its hydrophobic core, that is its non-polar core, is concealed from its surrounding aqueous environment, so that its hydrophobic core does not come in contact with the surrounding water or the surrounding aqueous environment. Now, a peptide chain of a protein often contains a single domain, whereas mostly made up of multiple domains. Let us consider or take the example of a protein, say an immunoglobulin protein. For an immunoglobulin, usually there are two peptide chains. One is called heavy chain and the other is called light chain. The light chain of an immunoglobulin, that is the L chain, is made up of two domains. One variable domain called VL and one CL, that is constant domain, called CL. While the heavy chain, the heavy peptide chain of a polypeptide immunoglobulin or immunoglobulin protein consists of one variable domain called VH and multiple constant domains numbered as CH1, CH2, CH3 and likewise. So doesn't it sound interesting? Yes, it really sounds interesting, studying about the way the different elements of secondary structure are grouped together to form super secondary motifs, which form the basis for higher order structure of proteins and classification, structural classification of proteins. So at this point, I'm going for a very short break. I am leaving an activity for you all. Think about the way the elements of secondary structure are arranged to form super secondary motifs. Just think about the different ways, or think about the various, that is, alternatives in which the elements of secondary structure can be arranged to generate the super secondary motifs. I'll resume back after a very short break with an answer to this. Till then, that is, keep on doing with this activity.